So our next speaker is Dr. E.V. Soniya, and she is from the institute where I am from. So she is Senior Scientist and Dean of Academic Affairs at Rajiv Gandhi Center for Biotechnology, Trivandrum. She is also adjunct professor at the Regional Center for Biotechnology, Faridabad, and her work involves genomic DNA fingerprinting, plant biotechnology, and metabolic engineering. And she focuses mainly on black pepper and to identify the characterization of type 3 PKS involved in the production of medicinally important natural plants. She has a number of awards which she has got, including the Janaki Ammal National Women Scientist Bioscientist Award, Best Women Scientist Award from National Academy of Biological Sciences, the Young Scientist Award, visiting professorship at the McGill University, Canada, Rajalakshmi Award for the Eminent Woman Achiever in the field of research in biotechnology, and she is also the elected fellow of National Academy of Sciences. She has around 49 publications, and today she is going to talk on the topic of plant biotechnology applications and challenges. So over to you, Dr. Soniya.
Thank you, Dr. Jackson, for the nice introduction. Thank you also very much, Dr. Shivakumar sir and Professor Ganga Prasad for inviting me for this very important meeting. And actually, my topic is importance of plant biotechnology and its application and what are the challenges. So as you know, the plant biotechnology is one of the very important division of biotechnology with the ultimate aim of to feed an expanding world population. As we know that agricultural, maybe since the emergence of agriculture almost 10,000 years ago, and started domestication of animals, plants, and then the civilized culture started developing. And but the major
just sorry for interrupting you. Is there a slide that you can project, or you just?
Yeah, it is there.
Yeah, please.
Oh, so I forgot to share that actually. It's okay now?
It's fine. It's fine.
Okay. So actually, even though we have, even though our agricultural practices are improving continuously, developing, but still the problem is actually we have productivity is not satisfying with the increased demand of demand, and because of mainly the availability of farmland and many other demands for renewable product, many other reasons. So the grand challenge in plant biotechnology is actually foremost increasing the productivity in the in a higher level. So we have to actually depend or achieve the quantitative improvements in food, and where the plant biotechnological biotechnological tech appears to be hold very much promise. But at the same time, even though compared to the human biology, plant biology is maybe a little bit tough, actually dealing with additional level of complexity that is encountered by the fact that plants usually are grown in constantly changing environment that are almost actually always far from optimal, and climate changes, and also many change in the ecosystems. And more than that, I don't know how many of you know that ploidy level, especially the plant I'm going to talk about, the black pepper, that is also very one of the difficult thing which we always face. So coming to my, so what I thought was actually, I will maybe describe the plant biotechnology, maybe its applications, mainly through the work which I have done maybe for the past few years, not few years, long years. So I started maybe initially when I was doing my PhD. According to me, the plant biotechnology was plant tissue culture. Later, I understood there are a lot of other maybe ways by which we can improve the plant through genomics. Many different approaches we can take. So the first actually, after joining Rajiv Gandhi Center for Biotechnology in 1997, the first work I have done is mainly the genetic analysis of somaclonal variations, mainly the callus region derived plants of tomato. So for that, actually, we have raised different maybe plants through different callus cultures and compared, and we analyzed maybe the diversity. That was one of the first work I have done in RGCB after joining RGCB, and for that I got a Young Scientist Award from the Kerala State in 1999. And also, since I have told you that in plant tissue culture I have a very good expertise, so I started with giving a project mainly to maybe for the multiplication of the maybe medicinal plants through in vitro method. So this was funded by National Agricultural Technology Project. And then after that, we moved mainly to the black pepper. The reason was actually Department of Biotechnology also wanted us to work on black pepper because of its importance as a spice crop. Even though the difficulties, at least whatever we get, the leads will be good for the maybe the farmers. That's the reason we started working on the black pepper. So usually, as I told you, I have told you that I have used the RFID marker for analyzing the plant stage through callus variants. So later we maybe started with working on FLP. FLP is a comparatively maybe better method to develop markers. So then we move to the SSR. SSR means single sequence repeats. The reason the SSR is the most actually we can depend on, especially it's supposed to be the markers maybe people now maybe depend on. And since the maybe black pepper doesn't even have the knowledge of anything genomic level, so developing the SSR markers was a tough actually job for us that time. And one of my first student, Nisha Joy, she has taken all the effort, even developing the FLP markers, data microsatellites, based on the enrichment maybe method we developed in our lab, that we have maybe enrich the microsatellite, then we sequence, then when we maybe the flanking, based on the flanking regions, we develop some design some primers and then validated in more plants. That is the way we have done developed some markers based on the microsatellite for varietal identification of black pepper. Just to show that we are very fortunate to have our Nobel laureate Watson to visiting RGCB maybe in 1999 or 2000, that period. And then another important thing actually that time our director, Dr. Ahmad Das, wanted was working in the maybe transformations, genetic transformations. So since I have a good expertise in tissue culture, again parallelly along with the markers, I parallelly started working on the transformation also. So first successful maybe transformation I got it in is cucumber. It's a salad cucumber where I could show that it can be transformed through any gene of interest. And then another interesting work I have done. You can ask me in between also, there is no issue, because these are all part of actually different maybe approaches one can take for improving maybe the plant or agricultural crop. So in this case, very interesting thing is we have introduced maybe surface antigen portion of the maybe hepatitis B surface, hepatitis B, and then we transform that into salad cucumber, and then we are producing the maybe the protein in the plant. So in maybe it's just to maybe showing that we can maybe produce vaccines or maybe the proteins maybe in plants in large scale, mainly for the concept of edible vaccine. So that was also successful. But a problem I can tell you that if you wanted to get more protein, and then you had to maybe go for tissue specific promoter, many other things you had to see. So this is just to show that it is actually working perfectly, and you can use that system as a like producing the edible vaccine or related things. And then along with that, the student Sindhu also did actually, she cloned and characterized food specific promoter from Cucumis sativus, so that this protein will express only in the fruit. So that was actually major interest. So these are actually, this work actually even funded by Kerala State Science and Technology for funding for expressing the hepatitis B surface antigen. Then another one was actually in ginger, and we actually, as I told you, we were mainly focusing on black pepper and ginger, developing transformation was a great challenge for us actually. And the reason I can tell you that because of the too much contamination, which and many other recalcitrance of the tissues. And again, DST has supported, DBT and DST has supported to develop some protocol in ginger and black pepper. And it was not that easy, but somehow we could achieve some the results, and it is shown that it is possible, but it takes a long period, and one has to be very patient enough for years to get this result. So with that background, I completely moved my work mainly to focusing two things. One is like plant pathogen interaction in black pepper. Other was identifying the novel type 3 PKSs for exploiting their potential using metabolic engineering. So as I told you, metabolic engineering is also a part of plant biotechnology. You can produce maybe the proteins or whatever of your interest in plants in large scale. So as you know, the black pepper, I can go a little fast, and in between, maybe if you have any doubt, I can stop and answer that also. So black pepper, we started with, as you know, it is a king of spices, and it is Kerala and Karnataka. Tamil Nadu is the center of origin of black pepper, and also one of the very important and earliest known spice produced and exported from India. And we also use it for our day-to-day life for many purposes. And also it increases the bioavailability of many drugs and supplements because of the pipeline content. And so our major maybe problem we are addressing is against the phytophthora capsaicin and oomycete, which maybe causes kickwell disease in a plant. It's a devastating disease, and until now there is no actually maybe there is no remedy for that. So we have addressed maybe almost all the aspects to maybe address to this particular disease. So initially, our major challenge to maybe is to generate the genomic data resources. When I started working on those, even a single maybe nucleotide submission for that. So so we started from the scratch. Even start maybe small sequences we started submitting in NCBI. Later now we have in a state that we even had a whole genome sequence information, transcriptome, many other, almost every maybe genomics part is almost addressed. And so we started working with DNA fingerprinting, as I told you that we have done with RAPD, FLP, and later STR markers. STR markers, the importance is we developed maybe black pepper specific and then worked on that. Now, then we have also worked on the understanding the role of defense genes. When you start working on plant pathogen interaction, this is very important that you should understand what are the defense genes during infection and what are upregulating what is happening to the plant. So all these things has to be studied. So that we have done. And also understanding not only the maybe defense genes. Again, there are a lot of maybe so many regulators which control or regulate the defense gene. So you should have an idea of that also. So for that, we have studied the non-coding RNAs, because now everybody knows that non-coding RNAs are very important and for the regulatory function. So, and again, so we have studied not only miRNA and maybe maybe small RNAs derived from tRNA, ribosomal RNA, even long non-coding RNAs, everything. Even epigenetics we have studied for any regulators in immune response. And parallelly, we have to understand the role in the plant immunity. Parallelly, we also studied the pathogen side, because we cannot maybe only look into the plant. At the same time, also we have to see what are the maybe how the effectors, all these studies has to be done. What are the microRNAs present in the pathogen which control the resistant gene of the plant? All these things, someone has to be well known before you address some issues. So we also studied from the pathogen side, and also we finally wanted to elucidate the mechanism of trans-kingdom small RNA movement, all these things. Finally, we had to have a knowledge and mechanism of interaction between piper nigrum and phytophthora capsaicin during quick disease for developing strategies for improvement programs. So when you study actually a particular problem, you had to see from all aspects, like So these are some of the data set initially, because in 2010, almost we have started working on transcriptome. I think black pepper, I can say that the first time we have done that black pepper transcriptome, everything. Then we have addressed from the fruit, every part of the plant. And even though that time it was too expensive, but it was actually really needed because when you study on such problems. Then phytophthora, infected, control, everything we have studied. And then we found that especially when you look into the defense genes, mainly we found that the NBS, LRR, and ST cases are most commonly expressing when the pathogen is infected. And lot of isoforms, you can see that NBS, LRR, 291 isoforms are there. It's a multigenic family, and ST cases 23 is there. And also we found that metabolic data also. So parallelly, we have done metabolomics also, and we found that this we have done with the help of University of Aberystwyth, UK, and with the help of Professor Lewis. And we found that we can see that elevated level of ABA production after the infection. So we found that there is a big role for elevated level of ABA usually function as a susceptibility factor during pathogenicity of black pepper. So these are the publications came in. And this is again initially we have done the comprehensive analysis of small RNA, transcriptome, everything. And we found that there are specific microRNAs which are specific and also targeting the disease resistant genes, and all we validated through secondary structures and all the maybe based on the target. And also, these are some other data from for example, how the small RNAs derived from rRNAs, how they are maybe cleaving the targets. And again, we found that another important thing that there is a unique signature at the maybe 5' end of the 5.8 is rRNA. And we found that this is also useful for the taxonomical identification, especially the families. And these are some of the publications based on that. Along with the, as I told you, there are different maybe families for even isoforms, and tRNA also have that type of thing. So it's very interesting if I specifically talk each and everything, but then that takes a lot of time. And recently we got a good publication in computational structural biology in this small tRNA related work also. This is initial results we got it published in Foundation Plant Science. And how that the target is validated, you can see that. And along with that, actually, now maybe Ashwati, the student, actually she's from Kerala University itself, from MSc. She actually studied the genome-wide analysis of black pepper tRNA genes and verifying the differential expression during capsid infection, and also the role of tRNA modifications and identifications of especially for critical stress response, and the function of the TRFs. TRFs means the small RNAs derived from the tRNA during Phytophthora capsicum infection. And another important thing, the students, Shweta was done, is mainly the key regulatory factors controlling the synthesis of bioactive compounds in black pepper. We know that very important compound is piperine. So this, how the maybe regulatory factors are controlling the piperine production in the plant. So mainly we studied phenylpropanoid pathway, and also we have studied the microRNAs which control the pathway genes and how they are regulating the piperine biosynthesis. And we got published in BMC Plant Biology. We have got almost 5 publications in this year, 2022. One is actually the understanding the relation of phenylpropanoid pathway and how they are controlling the production of the piperine. And another one, this also we got published in Foundation Plant Science recently. This is the role of long non-coding RNAs, and again how they are regulating this maybe miRNA and other things during this biotic stress in black pepper. They also control miRNA, long non-coding RNAs. And another one we have done also is from the pathogen side, and maybe how what are the maybe how the RNA pathway is functioning, and how the capsicum is capable of targeting the defense genes of the plant, and how that transcriptome crosstalk is happening. So that's another important thing. And also we recently started working on many aspects. One is especially the methylation, how the genes are regulated. And again, another one is like rhizospheric as well as root endospheric bacterial communities of maybe uh, plant, maybe the resistant and susceptible plant, how actually they contribute to the resistance, and also in the secondary metabolite content. So this is a very important part of work. And again, we have identified a little bit and how they can be used again to maybe control the disease. That is the most important thing. So that is what we are now doing that. And we have now enough data set in all the aspects, and even who now start working, they will get all the details on the NCBA submissions, which we have already done. So at present, these are the things we are doing, that is analyzing the role of epitranscriptomic N6-methyl adenosine in countering infection, and again, the exploration of key regulatory factors controlling the synthesis of bioactive compounds in black pepper, and investigating the metagenomic profile of piper to understand microbial impact on disease-resistant induction-resistant cultivars to susceptible cultivars, and exploring the small RNAs generated by Phytophthora capsicum during the countering for to uncover methods of reducing disease severity, and also exploring the small RNAs produced by Piper colubrinum and capsicum. So how they are interacting. And another very important thing is to study the effect of colonization by you may be, most of them may be knowing about this Peliforma indica. It's a well-known bacteria, and how they increase. We showed that this may be very much important in early flowering. Otherwise, black pepper takes long time to flower. But when you colonize this particular bacteria, within 60 days, the black pepper start flowering and developing fruits. And of course, again, another one is actually development of disease management strategies against Phytophthora utilizing biocontrol agents and green synthesized nanoparticle. Actually, this is funded by DST. So all these things are ongoing, and I'm not going to detail about the type 3 PKSs because it will take more time. And but the major things I can tell you that this is mainly to identify the novel forms of type 3 polyketase synthesis for exploiting their potential using metabolic engineering. So of course, the type 3 PKS is very important, and it can maybe, it is actually involved in the production of secondary metabolites in plants. So we can do the pathway engineering, and we for this we have developed a database, and we have identified enough novel forms and found that the product and how we can maybe increase the content, or how we can regulate that. And even with that, we can change maybe through site-directed mutagenesis. We can even go for maybe pathway engineering and to produce novel therapeutics. So these are some of the examples we have done in ginger, turmeric, bael, and gooseberry. And interestingly, we could show that in bael, we have successful in identifying the maybe QNS, quinolone synthesing type 3 PKSs, and we could show that in vivo, in vitro, everything. And also we can show that this is producing in vitro this product, quinolone product. And also then we have even the mutate site-directed mutagenesis. We could show that there is a change in the product maybe synthesis. And similarly in the gooseberry also we have done. So here after the site-directed mutagenesis, we could produce that maybe instead of quinolone, it is producing the flavonoids. But here in the gooseberry, we could even able to produce after site-directed mutagenesis the quinolones. It's a very interesting work, but that needs a lot of time, because I just wanted to show you the applications of plant biotechnology. That's why I'm going through a little bit earlier, maybe fast, but in between, if you wanted to ask, I can answer everything. And even we have worked in like, these are some of the publications we have got, and based on that, and when you overexpress this quinolone synthase, we could get maybe increased secondary metabolite in the plant. Even it shows more resistance to biotic and abiotic stress. And another very important work we have done is the crystallization. After the maybe protein we isolated, we crystallized the, and we got in native substrate bound and byproduct maybe bound. It's a very interesting work, and now we could even structure. Already we have maybe analyzed, and we have submitted to a very high impact journal also. And these are some of the related things, and how we are changing through site-directed mutagenesis. And even though this is modeling, we could show in through experiment also. So finally, my aim is to produce actually the product in vitro in large fermenters, and which are especially the secondary metabolites. So the major leads I can tell you that now in black pepper we are in a position that we have good leads. At least we have identified certain bacteria or certain things which we can PGPR having PGPR activity from the studies in rhizospheric and endophytes, and this can be used as a biocontrol agent against the Phytophthora capsaicin. That is one thing. And we also have now doing some CRISPR, especially from the pathogen and also in the maybe plant, also to produce the maybe the plant for tolerant to this disease. So we have now we have all the data. Only thing to maybe in the next stage, and producing a plant maybe tolerant to the disease. That is the maybe that stage we have reached. And in the case of polyketates also, we have reached in a level that we can maybe synthesize this product, maybe produce this product in large amount in the fermenter using the yeast expression system. So this is one of my very important actually conference I maybe got from the EMBO for the financial support. And this is the first plant actually EMBO conference in India. And of course, collaborators are very important because of maybe Dr. Rajesh Kagaya could able to complete the quinolones in this part, and Professor Vijayan for the crystallization and other thing. Use more for my two students actually, because Commonwealth Fellowship and Newton Baba worked in Iber's University with Louis Moore, and we did lot of metabolic that I couldn't present everything. Metabolomics with this help, and Debashri helped in Debashri Bandyopadhyay from BIS Pilani helped me in especially the crystallization, the data analysis part of the structure of the quinolone synthase. And of course, these are the funding agencies. Because of that only I could do work on this. And also I am very much thankful to our all directors from Dr. Yamar Das, Dr. Tampan, Dr. Radhashna Pillai, Dr. Professor Radhashna Pillai, and then the present director. Thank you very much. Open to questions.
Thank you, Dr. Sonia, for the wonderful talk. And now it is open for questions. We can take a couple of questions if it's there. I don't see any questions.
Maybe I was so quick and to complete. Maybe that may be the reason nobody have.
No, I think everyone is too hungry.
Why my video is not coming?
No, it's not coming up. Yeah. Yeah, now it's fine. Yeah.
Okay. Anyway, so, I'm happy to maybe share because I only told very, very less only, because it's not possible for me to do everything this time. My aim is to, what are the maybe the ways one can maybe do in plant biotechnology, maybe to address the issue. So that's why I covered almost everything, maybe in a quick way. And also finally we have done the whole genome also. So anybody who can work also will be actually very helpful also.
Yeah. So I think if there is no questions, thank you, Dr. Sonia.
Thank you very much. Okay.
Dr. Neetu, I think we can wind up now. And thanks to Dr. Ganga Prasad and Dr. Shivkumar for inviting me and Dr. Neetu to chair this meeting, this session of the meeting. So thank you all, and over to you, Dr. Neetu.
Okay, thank you very much.
Yeah. So, Dr. Neetu, you want to conclude?
Maybe just a quick wrap up of the session. I think it was a wonderful, today morning, it was wonderful to listen to topics ranging from virus gene therapy to omics for mass data analysis. And, and really interesting to know that vitamin B deficiency, B12 deficiency lead to dyslipidemia and CVDs in Indian population. And, Dr. E.V. Sonia's wonderful talk on a whole lot of work on genetic methods for improvement of pepper and spices and crops. It was difficult for me to assimilate a whole lot of information from her work. And then Dr. Krishna Kumar's effort on bioremediation techniques for to remove the perchlorate contamination. We both of us thank the organizers for giving us an opportunity to listen to wonderful talks and chair the session. And over to the organizer for further to proceed with conclusion or next session.
Thank you, ma'am. That brings us to the end of our invited talks on the second day of the webinar. We are extremely grateful to Dr. Jackson James and Dr. Neetu Mohan for chairing the session and summarizing the. Thank you both so much, and it has been wonderful to have you on board.
Thank you. It's been a pleasure to be there. Thank you.